Now, welcome. We are going to discuss on the process of cell division, and for now we are going to look at the introductory part of it. Now, by the end of this session, I'll expect you to be able to describe the structure and the functions of a chromosome, and then number two, you'll be expected to describe the process of cell division in a living cell, maybe a plant cell or an animal cell. Now, we will be starting the session by looking at what really happens in a cell. And in Form 1, we discussed about the structure of a cell. Now remember, the cell is the basic unit of life, whereby all processes of life occur. And one of the processes that occurs in a cell is the process of reproduction. So we cannot understand the genesis of reproduction unless we get into the nucleus of a cell. Now, in these diagrams here, if you look at it keenly, at the center, we have some thread-like materials, thread-like strands or thread-like filamentous material. Now, these substances or these structures are very, very critical when it comes to the process of reproduction. Now, these strands or structures that are thread-like, they are long, they are thin, and they are embedded within the nucleolus, are the structures that forms the basis of reproduction. Now, therefore, these structures, we refer to them as the chromosomes. Now, the chromosome is the genetic material. Actually, it's referred to as a vehicle of heredity, a vehicle of heredity. The reason being, it carries the genetic matter. Now, the genetic matter in this case is the DNA, deoxyribonucleic acid. Remember, the DNA forms part of the nucleic acid together with the ribonucleic acid. But the ribonucleic acid is little in amount inside the nucleolus or the nucleus, inside the nucleus. The amount of RNA or ribonucleic acid is lesser compared to the DNA. And I'm saying that the two of them are the ones that we refer to as the nucleic acids. Now, if you look at this cell here, it is membrane bound. It is bound by a membrane that we refer to as the nuclear membrane. A nuclear membrane. Remember, we have different types of membranes. The cell membrane here, the cell membrane. So in this cell, there are two membranes. The cell membrane that encloses internal contents of the cell or the structures of the cell, and then the nuclear membrane, which encloses the nucleus and the nucleolus. Now then, the genetic material which is carried in the chromosome is embedded within the nucleolus and then bound by a nuclear membrane. This cell is said to be eukaryotic. The cell is said to be eukaryotic. And for multicellular organisms, this is the type of the cells that we find in them. That is, the cells that have their nuclear material or their genetic material bound by a membrane. And remember, the membrane in this case is the nuclear membrane. Now, we also have another type of cell which has the nuclear material floating or freely within the nucleus, placed or positioned freely within the nucleus. And this is the type of the cell that we call prokaryotic. prokaryotic, as seen in the bacteria. Their cells are not membrane bound, and therefore such cells are called prokaryotic. But for those of multicellular organisms, for example human beings, we can refer to them as eukaryotic. That is membrane bound. The nuclear material, the genetic material, is enclosed using a nuclear membrane. So within the chromosomes, that is where we locate the genetic material. Now, the genetic material, in essence, we shall discuss them in greater length in Form 4 topic, genetics. Therefore, as of now, it's just a matter of overview and just a prior information that the materials that are embedded or that are found within the chromosomes are the DNA and sometimes the RNA. So we shall discuss on that in the topic of genetics. Now, if you look at the structure of the chromosome, number one, we say they are thin, long, thread-like structures. And these structures, they take two properties. Either they are observed within the nucleus of a cell, especially a cell that is undergoing division, remember, at prophase stage. So that means that we can only observe the chromosomes in a cell that is undergoing division, but by use of a powerful microscope. Otherwise, in most cases, they are observed as granules that are suspended within the nucleolus. Now, if you obtain a single section of a chromosome, you realize that it is made of a thin thread-like material that we call the chromatin. So the chromatin is the thin thread-like material that constitutes a chromosome. Now, these materials can be bound. A single strand of it can be bound with another at the center using a structure that we refer to as a centromere. Therefore, if you look at the structure where my cursor is here, you realize that there are two long coiled thread-like strands, and these strands are the ones that we call the chromatin fibers. Now, when they are bound together at the centromere, then we refer to them as the chromosome. And remember, it's only visible by use of a powerful microscope, and number two, only when a cell is undergoing a division. And in this case, such a phenomena or such a feature can only be seen in the prophase stage of division, either prophase 1, prophase 2, or prophase of mitotic division. We shall look at the two types of division later on. Now, along the strands of the chromosome is where the genetic material is housed, or located. Now, a specific part of the chromosome that you can be able to locate a gene, or a DNA for that matter, is referred to as a gene locus. Now, a gene locus contains particular genes that determine a certain trait that is observed in a given organism. Now, naturally, the chromosomes do not occur singly, but they occur in pairs. And the pairs are identical in form, shape, and structure, but only different in genetic constitution. Meaning that a single chromosome may have different genetic combinations. There could be a gene for a portion where the genes for eye color are located, a portion where the gene for sex is located, either X or Y chromosome. We shall see on the types of chromosome, and many others. Now, when the two strands are joined together, they form a structure that is uniform in shape, uniform in size, but different in genetic matter. Now, such a chromosome is the one that we refer to as homologous chromosome, a homologous chromosome. Now, in a dividing cell, once the cell takes up the strain, these structures are seen as in this diagram here. The long thread-like structures condense, they become thicker, and therefore they become visible. That's why you can now be able to observe them, like in this case. So, it is easy to say that a chromosome is made up of two sister chromatids. And the sister chromatids are the ones that forms the arm. Now later on we shall be told that there are long arms that are referred to as Q arms. The longer one are referred to as Q arms. In this case the diagram may not be so evident, but we need to know that these sets are the ones that are called the chromatids. So a chromatid from one segment is equal with the other of the contrasting pair. And therefore, the use of the word homo. Remember, the word homo means same. And along the length of the chromatids is where we find the position of the genes. And a specific locality of a gene in a chromosome is the one that we refer to as a gene locus, as I've given an example in the two here. One for gene for eye color, either blue, and the other one red. Now, some of the other features about the chromosome, you shall get them in the notes that I shall accompany to the video. Therefore, very quickly, let's look at this table here, that we have the summary of all the properties of the chromosome. Number one, I said they are long and thin, thread-like structures that are found in the nucleus of a cell. Number two, they are greatly and loosely coiled. Number three, they shorten or condense and become visible at prophase, that is during cell division. Number four, they consist of two chromatids. Remember, the chromatids are the ones that forms the arms. There are longer arms, there are shorter arms. The longer arms represented by Q, the shorter one by P. Now, next one, chromatids are held at the center by a structure that we refer to as a centromere. Then, the other property is that the chromosomes can separate, especially during cell division, mitosis, and in the second stage of meiosis, that is during anaphase and the anaphase 2 of meiotic division. Number 5 or 6 here, chromosomes takes most dyes and then they stain darker than anything else, and that's why they are referred to as chromatin. Actually, the word chromosome comes from, is an abbreviation of chromo, which simply means color. And therefore, because it takes color, we call them chromosomes. And then, lastly, they have fixed number per species. They have fixed number per species, as we shall finally look in this table. Now, you realize that Now, you realize that we've said that each organism has a certain fixed number of chromosomes, either in the body cell or the gametes or the sex cells. For example, for human beings, it is 46. Now, for the sex cells, it is halved in what we call haploidy to 23. Mosquito, 3. And then for the sex cell, it is halved, or haploid status is 3. So meaning that the body cells have twice the number of chromosomes, but the sex cells have half the number of chromosomes. Now, we shall look at the other areas of the chromosome later on in Form 4, but in Form 3, that is the most that we need to look at in terms of the structure and then the function. And then now when we discuss about genetics, that's where we shall deal about the chromosome and even look at the amendment of the DNA. Thank you.